I'm your host, Nick Jikomis, and today I'm speaking with neuroscientist Dr. Michael Crair. First, just some brief housekeeping. I had some technical difficulties with this episode. I was not recording my audio when I was speaking appropriately for about the first half, so the audio will sound a little off compared to what it normally sounds like, but you should still be able to understand everything I'm saying, and it should be relatively clear. So apologies for that. It goes back to normal about halfway through, and when Dr. Crair is speaking, it sounds like it always does. Anyways, I spoke with Michael, who is a professor at Yale. He's a professor of neuroscience as well as of ophthalmology and visual science. And Dr. Crair is actually a physicist. His PhD is in physics, but he became a neuroscientist and he runs a neuroscience lab. His lab studies brain development. In particular, they employ advanced imaging techniques and other techniques that they use to understand the basic mechanisms that mediate brain circuit development, how the brain actually wires itself up when an animal is developing in order to create a fully functioning, mature brain. And we talk a lot about research and topics related to what they study. We talked about learning and memory, different forms of plasticity, including something called cortical map plasticity and what maps are in the brain. We talk about critical periods of plasticity in development. And we also talked about some really interesting research that's come out of his lab recently where they show that in mice, before they're ever even born, when you look at visual parts of the brain in these mice, the brain actually seems to be simulating patterns of activity that the mouse will encounter in the real world after it's born and its eyes open. In other words, parts of the visual portions of the brain are displaying patterns of activity that look like the patterns that you'll see later on in the brain of that mouse after its eyes open and it's actually looking at the world and moving around in it. So the brain is doing this kind of simulation of the external environment in order to train or pre-train the circuits of the developing brain so that they are pre-prepared to deal with what the animal is going to encounter, which is pretty cool if you think about it. So if you're interested in brain development or learning and memory and things like that, this will be a great episode. As always, if you enjoy the content that is provided on the podcast, we really do appreciate 5-star reviews on Apple or your favorite podcast directory, comments, likes, and shares or downloads of the podcast, either the audio version or the video version on YouTube. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. This episode is brought to you in part by Mudwater, a coffee alternative made with extracts from 4 different mushroom species and a blend of all natural herbs. Mudwater contains around 1/7 the caffeine content of a normal cup of coffee, so you can get the energy you want in the morning without the same chance for the anxiety, the jitters, or the poor sleep quality that can come from drinking too much coffee. 2/3 of Americans just about are drinking coffee every single day, and the caffeine in that coffee has a half-life in your bloodstream of between 5 and 9 hours for most people. So if you're drinking into the afternoon or the evening, there's a good chance that caffeine is still in your bloodstream and it can disrupt the quality of your sleep. So if you're trying to drink less coffee or you think you might have a caffeine habit, and caffeine is absolutely a habit-forming psychoactive stimulant drug, then check out Mudwater. It has 1/7 the caffeine content of a normal cup of coffee coming from masala chai, and it also has cacao, turmeric, sea salt, cinnamon, and extracts from chaga, reishi, cordyceps, and lion's mane mushrooms. The other thing that's really cool about this company is they donate a portion of the revenue to MAPS, the Multidisciplinary Association of Psychedelic Studies. We've actually had people from MAPS on the podcast before talking about the research on MDMA-assisted psychotherapy for PTSD and their other work into research on psychedelics. So if you want to support that movement, this is one way you can do it. If you're interested in trying Mudwater, check out their website, mudwtr.com/mindmatter, and you can use the code MINDMATTER, all one word, for $5 off. That information will be in the episode description, so check it out if you think you're interested. Professor Michael Crair, thank you for joining me.
Cheers. It's nice to be with you, Nick.
Can you describe for everyone who you are and where you're calling in from today?
Yeah, you bet. So my name's Mike Crair. I'm a professor in the Department of Neuroscience at Yale University. I'm also the vice provost for research at Yale. And I'm calling from Guilford, Connecticut, which is a little suburb of New Haven, Connecticut, where Yale is located.
So what do you study as a neuroscientist?
I'm interested in brain development. I'm sort of fundamentally curious in trying to understand how the brain wires itself up during development. And that's a pretty big topic. It's a diverse topic, but I think it's a fundamental interest to just understanding brain function is to try to understand how we all get wired up over the course of development.
And what's your sort of ultimate aspiration? Why are you trying to answer the kinds of questions that you are asking?
Yeah, so of course it's a good question. I'm interested in how we think, right? I want to understand how we think, and that's a $6 million question, to use an old phrase. That's a hard one, and I think fundamentally what I decided was at least one reasonable way to try to understand how we think is to figure out how the brain gets put together in the first place. If we can understand sort of the mechanisms for wiring up the brain, that'll give us fundamental insight into how it is that the brain works, how we think. And, I do those studies by focusing on sensory systems, the visual system in particular, because it's a relatively simple system to understand how the brain wiring occurs over the course of development. So I want to understand how the brain works. Very deep, fundamental question. And I choose to study trying to understand how it gets wired up as the best entree into that fundamental question: how do we think?
Yeah, I want to talk a little bit about some of the structures and the parts of the brain that your lab studies. On your lab website, you've got the research overview page, and it says something like in the brains of animals, the sensory world is organized in regular neuronal arrays or maps. So what exactly are these sensory maps in the brain?
Yeah, so the easiest way to think about it is at least one simple way that many people can grasp easily is the map of the body surface. So in the brain, you have areas that respond to stimulation of the body surface, the sensory periphery, and neighboring neurons in the brain will respond to stimulation of neighboring sensory receptors on the body surface. And so this is basically a map of the body in the brain. So and the fundamental feature of those maps is that there's a preservation of neighborhood relationships. That is, neighboring sensory receptors are mapped to neighboring locations in the brain. That's a somatosensory system analog of a sensory map. The same sort of map occurs in the visual system, where neighboring positions in the visual world are mapped to neighboring locations in the brain. So it's a sort of morphed map of the visual world in the brain, where neighborhood relationships are preserved. That's what we mean by maps.
I see. So morphed map, is that why when people see that image of the so-called somatosensory homunculus,
Exactly.
It looks like a very warped human being with exaggerated features for some places?
Yeah, exactly. No, that's exactly right. So, and the morphed features represent basically the density of receptors on the sensory periphery. So those parts of the sensory periphery, like your fingertips, have lots and lots of receptors. So there's a dramatically enlarged representation of your fingertips in the brain. And in the visual system, there's an analogous morphing where the fovea, the center of your vision, has a very, very high density of photoreceptors. And so there's a dramatically enlarged representation or piece of the visual cortex, which represents or responds to stimulation of just the center of your vision, the fovea. So it's a morphed map so that the distances are not preserved, but neighborhood relationships are preserved.
So I'm going to ask a question that's maybe vague, but I do think it's interesting and important. Why would animal brains be organized in this kind of way? Are there other like biophysically plausible ways you could imagine the brain being organized? And why did evolution create this kind of structure with these kinds of maps?
So I think these maps are really just the starting point of neural computations that lead to say cognition, higher order cognition. It's sort of a basic building block or representation of the sensory periphery. A fundamental feature of the world are these neighborhood relationships, like neighboring locations on your fingertip or neighboring locations in the visual world. And so one of the initial roles of the sensory system in the brain is just to take the input from the sensory system and code it in a way which is then easily digestible for higher order processing. So these maps are robust in the initial stages of neural processing, and then they become quite complex and not obvious, and the mapping relations break down. So I think it's an efficient way of coding information about the sensory periphery. That's why they're common.
And a lot of the questions that I want to get at next will have to do with brain development and the extent to which structures in the brain, such as these maps, are self-organized and just sort of an unfolding of things intrinsically specified by the genome versus things that are shaped by experience and that are very malleable. So before we get there, let's start off by talking plasticity, generally speaking. So what is plasticity, and what are some good examples in the mammalian brain of plasticity?
Yeah. Okay. So the plasticity is the malleability or the ability of the brain to change its response, or in this case, in the case of maps, the representation of the sensory world based upon the pattern of sensory input. So, we're talking about, maps of representation of the body surface into the brain. That map is not fixed. That map can change over time, especially it can change dramatically during development. And so plasticity would be the changing of the wiring that would lead to a difference in the representation of the sensory world. So for instance, if you, for some reason, lose a finger during early development, so that, your middle finger, let's say you're in an accident, you lose the middle finger, you would no longer have input from that finger into the brain. And the map representing your hand would change over time, it's plastic, such that the two remaining neighboring fingers would come to be represented in neighboring locations in the brain, and the middle finger would no longer be represented. It would disappear. That sort of plasticity occurs in spades early in development. It's a remapping or rewiring of the brain to accurately represent the condition of the sensory periphery. That sort of plasticity is, as I said, very, very robust early in development and much more subdued, much more modest in scale as you get older and older, such that the maps become relatively fixed as an adult.
And what's the purpose of plasticity being at high levels, basically earlier on in development, and then going away? You often hear, especially in the news, plasticity used as a very positive. It's always good. Is that the case, and does that have anything to do with why plasticity sort of wanes over time?
So, the honest answer is we don't know. I can give you my hypothesis or my understanding of why the plasticity wanes over time. So what appears to be the case is that the plasticity wanes. So the plasticity remains robust in the adult in higher order cognitive areas. Like higher order cognition is the brain circuits responsible for cognition are very plastic in the adult, and that's what allows us to think and learn as adults. The plasticity is wanes in representations of the sensory periphery, the sort of input circuits. And then as we get higher up in the sensory and computational hierarchy in the brain, the plasticity is preserved in the adult. So the further you are in the sensory periphery, the earlier in development, that plasticity wanes and the circuits become fixed. So it seems to cascade from the outside in. So the plasticity remains robust in the adult for cognitive circuits, but as you work your way out to the sensory periphery, the plasticity gets less and less, and the circuits become more and more crystallized earlier and earlier in development. So that's it seems to be a common rule. The question is, why would that be? I surmise that you're like the neural circuit. You're like building a cognitive circuit, a cognitive house, and the representation of the sensory world, these maps, are the foundation. You need to have a representation of the world, and that provides a foundation for higher order cognitive, thinking, extraction of relationships and features of the world. And so you initially build the foundation. It's a little plastic as the foundation is developing and being born, and then it becomes fixed, so that the higher order circuits can faithfully rely on the representation which was built early in development of this external world. So I view it as those early plasticity is to build the right foundation. That foundation becomes fixed, and then the higher order circuits, which are very plastic, remain very plastic, rely on a fixed foundation, so it knows what the sensory world representation is. It needs some sort of a scaffold, some structure to trust that is fixed for the higher order representations to then act on.
Interesting. Yeah, that makes that makes a lot of sense.
And that's just a hypothesis. The way I conceptualize why there's plasticity and why that plasticity wanes over time, and it wanes more in some circuits than others. The plasticity wanes earlier and more dramatically in the more peripheral circuits. And then as you work your way up in the brain, in the sort of cognitive architecture, the plasticity is sustained higher and longer in the adult.
I see. So if you're looking at a pair of neurons in the brain, the level of plasticity will tend to depend on A, how far into development you are, how old the animal is, and B, how close to your eyes and your ears and the external world that circuit is. The further in this cognitive hierarchy you are, the more plastic the neurons will tend to be later.
That's correct. That's right. And it's actually true in reverse too. As you work your way out to the motor system, the neural control of muscles tends to be more fixed, but the motor circuits in the higher order brain are more plastic in their representations and their abilities to adjust their control of the motor neurons. So it's sort of on the way in and also on the way out.
Mm-hmm. So even though there's quite a bit of plasticity, even in these sensory maps earlier on in development, there is, my understanding is that there's a lot of self-organization that happens, basically a lot that's pre-specified and encoded in the genome. To what extent is that true in the sensory systems that you work on? And why would you want to have a fair amount of a sensory map or a structure in the brain sort of pre-coded, embedded in the genome, ready to go?
Yeah, so this is the kind of one of those major fundamental questions is, to what extent do you pre-code the circuit in the genome, and to what extent do you allow activity, plasticity, to self-organize the circuit? And this is phrased in various ways in different contexts. It's a little bit like the nature versus nurture debate, where nature would be the genome coding the circuit structure, and nurture being activity shaping what circuit forms based upon experience or the environment. And as is always the case, it's neither entirely one nor entirely the other. It's both nature or nurture that shapes the development of these circuits. And my background is a physicist. I'm trained as a physicist. I got my bachelor's and PhD in physics from UC Berkeley. And my PhD was studying dynamics, nonlinear dynamics. And there was a kind of intrinsic structure. Dynamical systems have this intrinsic structure, which is self-organizing. And in chaos theory, you talk about attractor networks and attractor states, and there's sort of the forces or nature of those systems lead to order in an internal self-organizing way, a self-organizing system. And so I was attracted to try to understand how the brain works, insofar as it develops as a self-organizing system. It has analogies to self-organizing systems that have been studied in physics. And so when I learned about plasticity in the developing brain as a graduate student, I realized there was a certain parallel to these self-organizing systems in physics, where the plasticity and the role of sensory experience in shaping the connections would lead to accurate and efficient representations, could lead to accurate and efficient representations of the sensory world as a computational convenience. And so It is absolutely the case. This is a long-winded answer, but it's absolutely the case that both genes and the environment are important in the development of the nervous system. I'm less interested in the role that genes play because I don't think they really tell us about how the brain functions. I'm much more interested in how activity, sensory experience, shapes the development of the circuits, because that self-organization, I think, gives you fundamental clues about brain function. How is it the brain organizes itself? It's a self-organizing system, really teaches you about how the brain works. Whereas the genes, the genes are the genes. They doesn't really tell you about function.
Interesting. One of the interesting phenomena in the brain that a lot of people may not be aware of is the idea that neurons are spontaneously active. Everyone, I think, has an intuitive understanding that, when something happens, eyeballs see something, neurons in the visual system light up with some kind of pattern. But even in the complete absence of sensory information, neurons are active. Can you describe what spontaneous activity is and why it's maybe interesting to think about in terms of self-organization?
Sure. So one of my earliest discoveries, and this discoveries made by colleagues in the field, was that much of the brain appears to brain circuits in the visual system, again, is where we've studied this in greatest detail, appears to self-organize before vision. That is, many of the fundamental circuits that are responsible for representing the visual world, like location of stimuli in space, or orientation of edges, or direction of movement of objects in space. The circuits that are responsible for those specific response properties to location and direction and orientation form before vision, before the animal has seen. And so that suggested that there must be genetic mechanisms that are responsible for the formation of the circuits, or that there was activity in the system that was present prior to vision that helped the circuit reorganize. And again, me and colleagues demonstrated the presence of this early spontaneous activity which is actually generated in the nervous system prior to vision, that helps organize or train the circuit to form these sophisticated circuits for locating the location of objects or the direction of motion of objects in visual space. So it's activity that's intrinsic to the nervous system, that's not stimulated by outside light. And that early activity, this spontaneous activity, is important for developing particular circuits in the brain. And an analogy which I think is apt and appropriate is the kind of activity, dreaming activity. What I refer to dreaming. Nobody is surprised or shocked that everybody dreams. Essentially, that dreaming activity is spontaneous activity in your dream state. You go to sleep. It's not dictated by sensory stimulus. It's activity which is generated in the brain, which resembles some activity you had during the day. It might reflect some experience you had in the past, or it might not, or it might be completely imaginary and fictitious. But it's a pattern activity in the brain which gives some sense, some sensation, has typically visual properties, or might have olfactory or somatosensory properties. This is spontaneous activity in the brain, in the adult brain. The developing brain has spontaneous activity too. It has its own intrinsic pattern of activity, which we and others have shown is important in the wiring of the brain itself. The self-organization of the brain during development is actually dependent upon this early spontaneous activity that's completely separate from sensory experience.
Interesting. Michael, apologies. I'm going to pause real quick. So that's interesting. What you're saying is, we all dream every night. So we actually have direct experience of this kind of thing. The brain can simulate the world even when it's not receiving inputs from the world. On the other hand, I could imagine when I go to sleep tonight, I'm going to dream these kinds of simulations will be, playing some story in my mind's eye. But I could also imagine that, even though the sensory input has been turned down while I'm sleeping, all of it's been informed by previous experiences I had while waking. And so in some sense, maybe the information got in there from the outside world and I retained it. But you've also done research showing that this type of thing happens even in the developing system of a mouse before the mouse is ever even awake and opening its eyes in the world. So
right.
Tell me if this is accurate. What you're saying basically is, in mice, in the case of your lab, you can record from neurons in the visual system or look at them using a microscope, record them using an electrode. And the patterns of activity that you see in the developing nervous system before the mouse ever opens its eyes look like they do when the mouse will later be opening its eyes and experiencing the world visually.
So that's not quite right, but it's close. So the difference is that it's hard to. First of all, so let me back up just a second. We definitely see activity in the young mouse before it opens its eyes, actually before the retina is functional. So there's a robust spontaneous activity in the developing eye and brain before vision is possible. The retina doesn't work. And what we and others showed was that this early, what we refer to as spontaneous activity that's generated in the retina and propagates up to the brain, is essential for the normal development of the visual circuits. So in the absence of this spontaneously generated activity in the retina, neural circuits don't form correctly in the visual system. What we further showed, which is I think most remarkable, is that this early spontaneous activity isn't random. It's not random firing of neurons in some kind of sort of salt and pepper fashion, or like fireworks or something like that. It doesn't have it's not random in its feature, but it's actually highly patterned. And the remarkable thing is that this early activity in the retina resembles the activity that would be present if the animal was running forward through space. So it's a pattern activity which resembles the sensory experience the animal would see if it was running through the forest forward in space, not turning around in a circle, not moving backwards, not stationary, but actually it resembles the pattern of activity in the retina that would be generated in the retina if the animal was running forward through space. And this pattern of activity propagates from the retina up into the brain circuits, including the lateral geniculate nucleus, the superior colliculus, and the visual cortex. So all the way up the visual hierarchy in mice. So I can't say whether the animal sees something in a dreamlike state that resembles as what it would, I can't interpret what the animal sees, that it looks like, it seems like to the animal like it's running forward through space. What I can say is the stimulus, the activity that's generated in the retina and that propagates up into the brain resembles the activity that would be present if the animal was running forward through space. And so it's just an important distinction. It's a little bit of a fine point, but I think it's an important distinction. I can't get in the brain of a mouse and know what it sees, but I can. The activity patterns that are present are similar to the activity patterns it would see if it was running forward through space.
So it's as if the brain is more or less simulating the type of activity that's going to commonly come from visual experience in the world, which will often be, it is basically the idea that a common of moving through the world for the mouse is moving forward. And so that direction of motion is maybe the one that the brain is working on, pre-coding or pre-working on before the animal actually ever sees anything.
That's exactly right. And I think it's I think it's an evolutionary phenomenon. In other words, if you go back in evolution, animals like some primordial fish or amphibian or whatever the primordial beast that the mammals came from we're swimming forward through space or moving forward through space. And mammals have had evolutionary adaptation that leads to a very extended gestation. So the time in the womb is very long in comparison to, say, fish and frogs. And so during this, a fish and frog, hatches as a tadpole, has a very primitive nervous system, and it continues to develop over many years. Many months. And that primitive nervous system has continuous wiring, continuous formation of circuits from the eye to the brain in the presence of visual stimulus. And for a fish or a tadpole, it's swimming forward through space, and so it's seeing stimulus come by it. And under normal circumstances, or in the primordial circumstances, the animal, as its visual system was getting wired up, would have present this continuous or a relatively continuous stimulus moving forward through space, so the world coming past it. Now, since mammals have an extended gestation, they're obviously not seeing the world that way. Actually, the circuits are forming prior to the animal can see. And it's my hypothesis that it substituted it substituted a generation of spontaneous activity in the retina to mimic the kind of activity it would have seen in the sort of primordial soup as it was swimming forward through space. So it's mimicking the stimulus which was typical of what an animal would see with a shorter gestation earlier in evolution by this generation of these spontaneous patterns of activity. And that leads to the formation of circuits which are appropriate for things like the detection of motion through space or neighborhood relationships, location of objects in the visual world. I hope that makes a little bit of sense, Nick.
Yeah. Why would spontaneous activity be observed in neurons in the first place? Is this something that is baked in on purpose, quote unquote, by evolution, or is it just sort of like an inevitable noise artifact that comes from being cells?
Yeah. So I think cells, neurons, have this feature that they don't want to be silent. They have these what's referred to sort of homeostasis. They don't want to be too active, like firing all the time and going crazy, but they don't want to be silent either. If they're firing at high rates all the time, then they're burning out. If they're completely silent, then they're not doing anything. They're not being useful. So they have what appears to be a way to self-regulate their activity. So to regulate, for instance, the ion channels in the membrane to maintain some basal level of activity. And so they're sort of spontaneously active naturally. And if you take away, for instance, their input, they'll ramp up their intrinsic membrane properties or the strength of their synapses to restore some level of activity. So there's a kind of homeostatic mechanism to maintain a happy level of activity. But we're talking about here is much more than that. We're not talking about individual neurons having a happy level of spontaneous activity, but actually patterns of activity between large cohorts of neurons that are patterned, right? It's activity amongst large cohorts of neurons which have specific spatial temporal patterns, not individual neurons, firing randomly with respect to each other. So the neurons themselves have this homeostatic tendency to be active. And then in addition to that, during development, what we showed is that there are communication mechanisms between the neurons that lead to coordinated patterns of activity, not just random firings of individual neurons.
And so when you're doing these experiments in the lab, can you try and paint a visual picture for people of how you actually observe these patterns? What do the experiments actually look like?
Yeah, so we make use of modern optical imaging techniques and genetic expression techniques that allow us to visualize with light the activity of neurons. And the way that's done now commonly is you use gene expression techniques to make neurons express a green fluorescent protein, this a protein that's derived from jellyfish originally, that actually has been modified to act as a activity sensor. It's called a GCAMP. Okay, it's a green fluorescent protein which is linked to a calcium sensor, which is a measure of neural activity. So when the neurons are active, when they're firing action potentials, they glow brightly. And when they're inactive, when they're not firing action potentials, they do not glow brightly. So what we do is make the neurons of our interest in the visual system express this glowing protein, GFP, linked to the activity indicator, GCAMP, so that when they're active, they fluoresce brightly. And with a microscope, by actually, all we need to do simply is peel back the skin above the skull, because we can't see through the skin, but you can see through the skull to the brain. The fluorescence is so bright that, especially in young animals, you can see right through the skull to the brain. And when the neurons are active, they light up like a Christmas tree, and you can see the activity with a microscope, with a pretty standard, simple microscope. So we're visualizing the spontaneous activity in the brain through this optical technique, using microscopes and these genetically encoded activity indicators.
So when you say that you see these patterns of activity, that's a very literal statement. You literally have tricks that allow you to watch the neurons actually light up.
That's right. That's exactly right. It's not just, it's not a figurative see. It's a literal see. They are fluorescing. They're glowing when they're active. And we can see that glowing activity.
So how do you think these, so where sort of in the brain are you observing this? Is it relatively close to the retina? Is it higher up? Is it these sensory maps, or where is it in that hierarchy?
Yes, right. It's one of these sensory maps. Actually, we can do these experiments anywhere that's accessible by light. Okay, so one of the challenges, if you're, deep in the brain, the light doesn't penetrate, and so you can't see the neurons or the activity. So anywhere that's accessible by light, you can see. And in our experiments, the typical structures which we image is a structure called the visual cortex, the primary visual cortex, which is the entry point for visual information into the cortex, the cognitive part of the mammalian brain. And another typical visual structure which we will image this activity is called the superior colliculus, which sits at the roof of the midbrain, which is another important visual nucleus in mammals and vertebrates. So those are the two typical areas of the brain that we image this activity, the superior colliculus and in the visual cortex. And in both of these structures, there are maps of the visual world. So that, for instance, neighboring neurons in the superior colliculus will respond to stimuli in neighboring locations in the visual world. So there's one of these morphed maps of the visual world in the superior colliculus and in the visual cortex. So when we see activity in one location in the superior colliculus, we know that corresponds to what would happen if you were stimulating one location in the visual world because of this map.
So in some sense, you could watch the neurons light up in such an area and reconstruct what the animal might be seeing.
I in some sense, but I hesitate to draw that. I don't want to claim I can, step into the mind of a mouse and see what it sees. What I can tell you is the pattern of activity resembles the activity that would be present when you present this particular visual stimulus. So in some sense, you're right, but I hesitate to anthropomorphize too much.
So to what extent do you think this type of phenomenon is happening in the human brain? Is it maybe happening less because we have a more extended childhood and we're born in more of a state of helplessness compared to other animals? Or is this likely happening to some extent in the human brain as well?
I think it's happening in the human brain. So of course, the kinds of experiments we're doing are virtually impossible to do in humans. Some neural activity recordings can be done in infants using techniques such as functional magnetic resonance imaging, but it doesn't have the resolution to be able to observe these spontaneous activity patterns that we can observe in other mammals. So I don't know with any certainty whether it's happening in humans. What I can say is spontaneous activity in the retina happens in all other mammals for which it's been investigated. So other non-human primates, carnivores, and rodents, they all have this spontaneous activity early in development prior to vision. So I presume it's also happening in humans, but I can't say with certainty. So we know this early spontaneous activity exists. The degree to which it's patterned in a variety of mammalian species is a little bit uncertain. So the specific patterns that we've observed in mice. We don't know whether this early activity that resembles moving forward through space will be similar in other mammals, and that's something we need to find out. But we know for sure that there is early spontaneous activity. We just don't know what the details about the pattern in other mammals, and we actually know nothing about humans specifically.
Mm-hmm. So far we've established that all neurons are spontaneously active. In some sense, they're intrinsically built so that they're going to want to be spontaneously active at some rate. But not only are they spontaneously active, when you guys actually look at what they're doing in the developing mouse brain, you see that the activity is patterned. It has some kind of pattern. And remarkably, the pattern looks very much like the patterns that you might see when the animal is actually looking at something. How does the brain actually give it this pattern? What's physically happening so that it knows how to create these specific patterns and you don't just see like a random array of activity?
Yeah, so that's certainly one of the most fascinating things about this is in order to understand these activity patterns and manipulate them, change them to be able to determine what their function is, what their role is in brain circuit development, we had to understand the source and what system was responsible for generating the specific pattern that we see. So a lot of our work has been what I would call mechanistic work in dissecting the source and method by which the activity has this pattern nature. And so we know a lot about it. I don't think we understand it completely, but we know a lot about it. So one of the most interesting things is that not only are neurons intrinsically do they like to be active, but there are specific neural connections in the developing retina that form for the express purpose of generating these wave-like activity patterns that resemble the animal moving forward through space. What I mean is that they're not random connections that form during development. There's specific neural synapses, connections in the developing retina, which form in a pattern to generate these waves that resemble forward motion through space. They're developmentally controlled. So the circuits form for a specific epoch during development. For in mice, it's about a week. And then those circuits decay away and the activity patterns change and the animal has a visual function. So it responds to light instead of generating these patterned waves of activity. So there's a transient mechanism, a transient circuit that's formed during development, we think specifically to generate these waves of activity that mimic this forward motion through space for the purpose of training the visual system so that it can see once the animal eventually does open its eyes. So it's a developmental adaptation with a specific time point during development when the circuit forms and then dissipates and becomes something completely different.
Mm-hmm. Is it possible to actually test an idea like that where somehow you actually disrupt these patterns of activity before eye opening and then show that the animal's less able to perceive things like optic flow?
Right. Yeah. Yes, it is. And that's one of sort of an important technique is to test the the role of these early spontaneous activity patterns. They're sort of what I would refer to as their causative role. If you disrupt these activity patterns, what happens to the development of the neural circuit? So there's a very coarse way, a sort of sledgehammer way, which is to eliminate the activity completely, just basically completely block it. And if you completely block this early spontaneous activity, then the neural circuits that form are very perturbed. The animal's vision is very perturbed. It's sort of would see the rough shape and position of an object, but not be able to detect any specific features after it opens its eyes. If instead of just completely eliminating the activity, you change its pattern. So as I described, the early activity patterns appear to resemble forward motion through space. If instead of having activity patterns that resemble forward motion through space, you make it resemble random activity, sort of random motion. Motion, so there's patterns, but it's not a consistent pattern as if the animal was moving forward through space. We can manipulate the circuit so that it's this random pattern, and then when we look at the animal's neural response properties when it opens its eyes, what we observed is the animal doesn't detect, the neural system doesn't detect motion well. It has sort of primitive motion detection properties, but it doesn't do a good job of detecting motion. So by altering the pattern of activity in the developing retina, not completely eliminating it, but making it random instead of this forward motion flow, and then measuring the response properties of neurons in the visual system after the animal opens its eyes, we've shown that this forward motion through space activity pattern is essential for the ability of the animal to detect well motion once it does open its eyes.
Interesting. So there's a direct relationship between these patterns of activity and some. I mean, they truly are pre-training the circuits to actually do something.
That's exactly right. Because if you disrupt this early pattern activity and make it random, the circuit doesn't form normally, so that the neurons don't respond well to motion stimuli once it does open its eyes and it's able to see. So it really does appear to be essential for the development of the normal circuit for visual motion detection.
So this does, I mean, we mentioned dreams earlier. This does remind me of phenomena in the sleep literature where, you record from neurons in a particular part of the brain when a mouse or a rat is sleeping, and they resemble the patterns that you see when the mouse was actually going through a maze and figuring something out. So it's as if these simulations are happening to train the circuits even in the absence of the direct sensory input from the external world. Is that how you would begin to think about what's going on in a dream, say, something like that?
Yeah, that's exactly right. That's a wonderful analogy. So my colleague at Yale, his name is Professor George Dragoi, has studied these early activity patterns in the developing hippocampus, these sort of dreamlike patterns of activity, which we think are important for memory consolidation in the hippocampus. And what he's shown is that these pre-pattern activity in the developing hippocampus, these sort of dreamlike activity in the hippocampus at night, is necessary for the consolidation of hippocampal memory, this place. The hippocampus is responsible primarily for a place that has this a memory of where you are in space. These place cell memories are in part driven by dreamlike activity after the animal is done with a sensory experience. And between one day and the next, at night, in these dreamlike states, this pattern activity is necessary for consolidating its memory of location in space, which it saw the previous day. So this sort of pattern dreamlike activity is a pretty direct analogy to what we're observing in the developing visual system. It's not responsible for memory. Instead, what we're saying is that this early dreamlike activity is mimicking a stimulus, which is essential for the development, the self-organization of the circuit, so that when the animal opens its eyes, it's able to see. Instead of consolidating a memory, it's consolidating a circuit that's responsible for vision when the animal opens its eyes.
So I want to talk about human brain development for a little while. So humans, as I mentioned earlier, and I think everyone's aware of this, humans basically, as far as I know, we have the longest childhood of any animal, and we're generally the least developed animal and the most helpless at birth compared to most other mammals. And more or less what this means is we've got this sort of super wide window where there's a lot of plasticity and a lot of room for learning. And the flip side of that is maybe that there's sort of less baked into our brain compared to other animals. So how would say a human and a mouse compare in that respect? Is it actually true? And how much of the human brain would you say is shaped by experience versus how much is already there preformed?
Your description, I think, is pretty accurate, and the I think what happens in the human brain is very similar to what happens in a mouse brain, except there are obvious differences in the human brain. And the obvious difference is there's been a dramatic elaboration of the cortex. So, the frontal lobes and the parietal lobes are dramatically enlarged relative to a rodent. And so the difference in gestation and brain circuit development and plasticity is related more to the difference in the evolution of the brain than it is differences in in how the brain develops. What I mean by that is, I think the circuits that are responsible for the sort of in the superior colliculus and the visual cortex, the entry level circuits, and the role of this early spontaneous activity, and then the fact that the development is plasticity is so robust early in development and becomes more fixed, is true in the human as well, except for the human has so much more brain that's more highly evolved, sort of been added on relative to the mouse. And those circuits tend to be much more plastic and have longer developmental periods associated with them, and show robust plasticity in the adult. So I think there's really strong analogies, very similar, except the fundamental difference in the human brain and a rodent brain is that we have so much more higher order cortex, and that stuff is really plastic and has very prolonged gestation or prolonged development.
I also wanted to ask you about the concept of critical periods. So the one that is probably most commonly learned in school is maybe what you hear about in a psychology class when you talk about language development in human children. But there's also critical periods in developing sensory systems in mice and other animals. So can you define what a critical period is, and maybe start by talking about what we know from critical period studies in the visual system of mice?
Sure. So, critical periods are an important concept in understanding brain circuit development. They're sometimes misunderstood because they're thought of as open and shut. Like there's a door that opens and then a door that closes. And I think it's much more soft than that. So there's a roll on and a roll off, and the plasticity doesn't completely disappear. It just becomes much, much less robust. So with that premise, the concept of a critical period for the visual system in a mouse is very similar to a human. And so the example and so I'm going to actually use the human as a demonstration of the visual system critical period. If in a human you're born with one eye which is occluded, it might be because you have a physical damage, say, to the eyelid, so the eyelid is droopy or closed. You might have a congenital cataract in that eye, so the vision is blurry, it's opaque because of the thick of the optical effect of the cataract. If you have that visual, so you have trouble seeing through that eye early in development, if that physical trouble seeing through that eye persists after a few years, you lose the ability to see through that eye. And this is dramatic and easily demonstrated by you can restore the vision in the eye. For instance, you can fix the physical damage to the eyelid or fix the cataract. And this is actually commonly done now in adults. So adults develop cataracts, often they're age related. You can have a cataract, you can develop a cataract as an adult, have it for decades, and then have the surgery to repair the cataract so the optical properties of the eyes are restored to normal, and you can immediately see through that eye as if you never had lost vision in that eye. Whereas if you have a cataract as a baby, you're born with that cataract, or you develop it very early after birth, unless you fix the optical, of the eye, within a year or two, you will lose the ability to see normally through that eye. No matter whether you have years or decades of normal vision, if you don't fix the eye to normal working order early, you lose the ability to see through that eye. And so that's a critical period for visual circuit development, which is in this case is the first couple years after birth in a human. So this has had a fundamental effect, for instance, for how we treat vision deficits in babies. So if a baby is born with some vision, it might be a congenital cataract, or it might be the eyes are deviating so that they're looking in two different places at one time. This is sometimes called strabismus. You want to fix the physical and optical properties of the eye so it's normal, so that the neural circuits in the brain get wired with normal visual experience. And if you don't fix it right away, within the first couple of years, you lose the ability to see normally through the deviated or the cataract eye, even though optically you've completely restored it. So that's a fixed critical period for the formation of visual response or visual behavior. And the same thing happens in a mouse. It's got essentially the same critical period. So if you blur the vision through one eye or you deviate the eye, then you lead to abnormal vision in the deviated eye or the blurred eye. And that critical period exists in the first couple months after birth for a mouse. And by the time you reach about 2 months after birth, then that critical period is gone, and deviating or blurring vision in a mouse eye has much more modest or no effects. So the mouse visual system and the development of the neural circuits for visual response properties in that context is very similar to a human, although the timing is completely different, just because development occurs over a different time scale.
Mm-hmm. So once those critical periods have passed, even though there's not like one day where they sharply turn off and it's this sort of slow roll in and roll out, is it possible to open up some of those again pharmacologically, say?
Yeah. So of course, we and others are working hard now to identify pharmacological or genetic or other interventional techniques to restore critical periods. And there's been some significant progress in understanding what shapes the time course of a critical period, and in producing some restoration of plasticity in the adult. But we haven't cracked the nut yet. So, for instance, it appears to be the case in the visual system that the development of inhibitory circuits that are mediated, synapses are mediated by GABAergic synapses, inhibitory synapses in the developing visual cortex. The development of those circuits is really important for shaping the time course of the critical period. And if you delay or prolong the development of those GABAergic circuits, you can prolong the time course of the critical period. And there's some hope that if you could reintroduce some of those GABAergic synapses or those GABAergic neurons in the adult, you might be able to reintroduce a critical periods in the adult. So there are a number of attempts to identify what changes over the course of the development to frame the critical period and reintroduce the developmental elements in the adult to restore the critical period plasticity. And I think it's very, very promising research, but it hasn't. We haven't cracked the nut yet. It's probably multiple factors that change, and we may need to adjust to reintroduce multiple factors in order to fully restore plasticity, critical period plasticity, say in the visual system in the adult.
So one of the things I also wanted to ask you about was the relationship between plasticity and the direction you give it through experience, particularly in the context of something like a neuropsychiatric disorder. So, for example, a lot of the guests I've had in the podcast have talked about things like SSRIs, or more recently, some of the newer results with psychedelics, where it seems in general like what's happening here is some of these drugs, some more than others, induce heightened levels of plasticity that persist for some amount of time. But the plasticity itself is not intrinsically a good thing. It needs to be directed in one way or the other. So the simple idea here would be that this is what psychotherapy does. It's providing direction, whereas the drug is providing this kind of permissive signal for plasticity. Do you think that's a fair way to think about how drugs that induce plasticity, we should think about them, or is it possible that just inducing or shutting off plasticity can be intrinsically beneficial?
I think in some circumstances, introducing the plasticity can be intrinsically beneficial, but if you draw on the example of the self-organizing nervous system that we see over the course of development, if the self-organization is misdirected, so the activity the activity that's present when the plasticity is reintroduced is abnormal or leads to self-organization of a perturbed circuit, it's not helpful. And so not only should one reintroduce the plasticity, but the activity that is present, the training or the therapy that one might undergo when you introduce a plasticity has to be appropriate in order to train the circuit to a more functional state. So I think the analogy or the approach that you described is an accurate one. You have to be careful when you introduce plasticity, because you might end up actually destroying or disrupting the circuit rather than moving it towards a more functional state. So let's go back to the analogy that I brought up at the very beginning. If you have a foundation and you have a cognitive circuit which is built on a foundation, if you introduce plasticity into the foundation, and because maybe there's a piece of the foundation that's broken, if you introduce plasticity in the foundation and the plasticity is such that it just falls apart, it melts away, it doesn't do any good to the cognitive circuits. They'll also melt away, and they won't function well as well. They won't function better either. So if you're going to reintroduce plasticity into a circuit, especially in the foundational circuits, you better make sure that the activity that's present helps the circuit organize or reorganize itself in a way which is functionally productive. And that's the sort of cognitive therapy which one might undergo, or training, sensory experience training or motor training to help the circuit form in a productive way instead of a destructive way.
Yeah, I think that's an important point. I liked how you put that you could have a drug induce a heightened state of plasticity, and that can be directed towards a positive outcome, but it can also be misdirected if it's not being handled appropriately.
Exactly. Yep.
So one of the things that you always hear people say, and the number always shifts a little bit, and I feel like the number over my lifetime has always gone up, is that the human brain isn't fully developed until whenever, age 25 or something. And this is typically in the context of, talking about some social issue or policy decision. We should or shouldn't allow people to do things at a certain age because the brain isn't fully formed until some point. Is that true? Does it make sense to even talk about like an approximate age when brain development is over? Is it ever over?
Yeah. So, it's a very thought-provoking question. So, I like to think that we never stop developing. I agree with you that what we learning in the adult is just an extension of the developmental plasticity. So what may be what maybe another way of saying it is that if we go back to this critical period distinction, it's like the circuits that form are most malleable in certain epochs during development, and actually those are real epochs. So if it's too early, the circuit is not malleable. It's not subject to plasticity at all. And if it's too late, it's much less malleable. And I think the sort of cognitive circuits that you're referring to that develop in the frontal cortex, particularly in the 20s, they're most malleable in the 20s. That's when those circuits are forming most robustly and are most plastic. And then the plasticity reduces to a degree in the adult. And so it never completely goes away. And I would say the adult plasticity is like a continuation of a developmental phenomenon. It's just the robust plasticity is the most robust plasticity has already passed it by the time you get, say, into your 30s. So I agree with you. I think it's maybe inappropriate to talk about an end to development, because one can view adult learning and plasticity as just a continuation of a developmental phenomenon. And it's just the latest and most robust plasticity you see in the human brain is for cognitive circuits, which are the things that form in the 20s, which lead to things like planning and the ability to project yourself into the future and anticipate future events. And those sort of cognitive circuits are most plastic and form most robustly in your 20s.
Do you think that, stepping away from some of the hardcore science for a moment, a lot of people have commented on what I'm about to describe, but do you think it's likely or plausible that as we've developed new technologies, and as even something like COVID, where you've got children now who are not going to school for the lengths of time that they were, or maybe they're taking a year where they're essentially homeschooled, you've got things like our cell phones, which are so mesmerizing that we're spending not necessarily less time interacting with humans, but certainly we're interacting with humans in a different kind of way. As we achieve more and more technological process and technology comes into our lives more, is it plausible that the way we interact with technology is shifting the way that the brain is developing and shifting some of these epochs of development, such that the way that the kids are coming up today or developing today is literally different than the way it was, say, for you in terms of brain development?
Yes, I think it is. I think it's likely the case, but I don't think it's shifting the time course necessarily, but it's shifting the outcome. So maybe one way to think about it is the kind of typical experience, the sensory and social experience. Children now participate in is different than what I did as a child, right? We didn't have cell phones, and TVs were something you watched for an hour or two. You didn't have the ability to watch a screen for this period of time that kids do now, and you didn't interact with a computer screen like kids do now. So I don't think those differences between what I experienced and what kids do now changed the developmental epochs. But what they will do, or what they could plausibly do, is change the outcome of the development of the time course, what happens over the course of development. So, the circuits that form, the social interaction circuits that form in particular, are probably different now than they were before, screens, because the kind of social interactions are different. But I don't think that's necessarily good or bad. It's different. I don't think one can say whether it's, one could argue that the circuit is adapting to the experience the human receives. So it's adapting in a evolutionarily appropriate way to the experience that a kid now has. But I think it is reasonable to argue that literally the typical brain that's forming is different now than the typical brain that formed 50 or 100 years ago, when technology was so very, very different. I wouldn't say it's worse, and I wouldn't say it's better either. It's part of our adaptability as a species. It reflects the adaptability of the species. And let me just make go out on the limb a little bit. One could argue that social networks have also changed. As our sensory, or as our experience changes, as our interactions change, and our interactions, for instance, are over the course of computers or the web, or social media, then it likely changes the kinds of social networks that form, which are, after all, networks of neurons, right? Our brains are networks of neurons, and social networks are networks of brains. So they're networks of neurons. So the social networks probably have been affected by the changes in the way we interact with our environment in ways which are very hard to anticipate or even describe well.
Interesting. Yeah, no, I agree that you can't automatically say that this type of change in the development of brains, given these new social and sensory inputs that we have today, is good or bad. One thing that I think you could say, though, is because the rate of change driven by technology increases over time, the delta between generations in terms of their brain structure is getting bigger. Like if you went back 1,000 years or 1,000,000 years to our ancestors, right, the social and sensory environment that you had compared to your grandparents was more or less identical. Whereas today, it's more or less guaranteed to be very, very different.
Absolutely true. Yeah, absolutely true. So the differences in experience and even differences in nutrition
Hmm.
are much more significant now, or over the last 50 or 100 years than they were, say, 1,000 versus 1,100 years ago. And so, one can anticipate. Yeah, you're right. And so there may be even bigger changes in our brain circuits that form as a result of those large changes in both the experience that people are exposed to over the course of their life, and also differences in our environment, like nutrition and and other things like daylight versus nighttime. We're not exposed to circadian rhythms as uniformly now as we were 100 years ago when or 200 years ago when there was less artificial light. So there are a lot of things that have changed dramatically. It's hard, but yeah, and it's a great point. Hard to study.
Mm-hmm. So in general, as a neuroscientist who's been studying brain development for so long, how has your understanding of brain development influenced your general perspective on how you manage your own brain and mental health as you age?
So yeah, actually, interestingly enough, I think what the biggest impact on brain and mental health, the way that you can most positively impact your brain and mental health is through relatively straightforward means of having great nutrition, exercise, and sleep. And those are very, very fundamental behaviors which hopefully anybody can improve to sustain their mental state, their healthy brain state. Exercise, nutrition, having good nutrition, and good sleep. And I guess my work over the course of the last few decades has maybe it hasn't fundamentally changed that point. I think what my work and many colleagues' work has done has perhaps shown what the effects of exercise and good nutrition and and sleep have on the brain. But the fact that exercise, good nutrition, and sleep are fundamentally the best ways to preserve your brain function is it's simple, but it was true and understood 50 years ago as much as it is today. I mean, the secondary effect, I would say, is that things like continuing to be involved, intellectually involved. So a variety of reading and doing puzzles for fun and interacting socially with people, that sort of constant or ongoing stimulus is essential for preservation of healthy brain function. I actually think that those are secondary effects to the sleep and exercise and nutrition that I described earlier. But it's clearly the case that healthy brain function also depends upon healthy brain activity, and that is the sort of the plasticity or sustenance of plasticity. Sustaining good healthy circuits is in part dependent upon continuing to activate those circuits in a healthy way.
Yeah, it's amazing how no matter how much we learn, how much you read, or how much advice you ask, it really just does boil down to the most basic aspects of life: good food, good sleep, and good interactions.
Yeah, that's right. Again, I think that probably goes. There's a strong evolutionary reason for that. Um, but beyond those basic fundamentals, exercise and sleep and food, you can do a lot to sustain a healthy brain. But don't ignore those fundamentals.
Well, Michael, thank you for your time. This has been fascinating. Are there any final thoughts about brain development or neuroscience generally that you want to leave people with?
Yeah, I mean, I think this is an amazing time to be a scientist. It's in particular an amazing time to be a brain scientist, a neuroscientist. The tools and techniques that we have to dig in and look at what brain activity and manipulate to understand brain activity and brain development are amazing, and they're expanding all the time. And our knowledge of how the brain works and how the brain develops is expanding dramatically over time. I think there's lots to look forward to, both in our understanding of the fundamental mechanisms of brain development and the prospects for doing things like restorative regeneration of neural circuits or behavior, if we can understand the basic mechanisms of brain function and brain plasticity and development. It's a really exciting time to be a scientist, and in particular, it's a really exciting time to study how the brain works, how the mind works.
Dr. Michael Crair, thank you.
Thanks, Nick.